RNA world accelerated by dissolved iron 2.
It is believed that life originated with RNA-based genetic and metabolic system. For example, the RNA world. RNA was replaced by proteins after the creation of oxygen, leading to the current composition of our biological world. Today's RNA use magnesium ions as catalysts because they allow RNA to fold and function as an enzyme. However, RNA enzymes formed by Mg2+ are very slow. Fe2+ could be a mimic of Mg2+ in the absence of oxygen because of similar chemical properties. Some scientists suggest that it might be possible for early life to use Fe2+, which was abundant in Archean time while oxygen was not. Because of iron's atomic property, iron ions could potentially make RNA more susceptible to reaction. It is suggested that RNA ions could have been used for RNA prior to the Great Oxidation.
3 independent experimental systems were ran to observe RNA folding. For the first experiment, it's important to know that it's predicted that RNA conformation is conserved if iron 2+ substitutes for site-bound magnesium 2+, which is proven true in first shell RNA metal complexes. The RNA magnesium 2+ clamp and RNA iron 2+ clamp are nearly identical as determined by the density functional theory, which is also DFT. There are only very subtle differences, as you can see in the picture. This was determined by quantum mechanical calculations that showed that RNA conformation and coordination geometry are conserved when magnesium is replaced by iron in first shell RNA metal complexes. After making these calculations, they need to be experimentally tested. Basically, we're experimenting to evaluate iron 2+ in the absence of free oxygen as a replacement for magnesium 2+ in RNA folding and catalysts. In the chemical footprinting experiment, we use SHAPE, which is an acronym for Selective 2-Hydroxyl Acylation Analyzed by Primer Extension, as the technique used to predict and confirm secondary structures of RNA. For this, benzoyl cyanide was used in SHAPE to determine nucleotide reactivity, thus determining the secondary structure. The SHAPE was first used on sodium by itself, giving the reaction pattern a control group to compare to magnesium and iron, as seen on the graph. There are very many similarities in magnesium and iron with similar peaks at the same nucleotide positions. The pattern of SHAPE reactivity for the specific domain of RNA observed, which was P4-P6, is conserved when magnesium 2+ is replaced by iron 2+ under anaerobic conditions, which are possible early Earth conditions. This is when there was abundant soluble iron 2+. In theory, iron 2+ and RNA could support various groups of RNA structures and catalytic functions more diverse in RNA with magnesium as shown by this experiment.
In separate experiments, 2 ribozymes, L1 ribozyme ligase and the hammerhead ribozyme, were tested in the presence of Mg2+ and Fe2+ at room temperature and anaerobic conditions. For L1 ribozyme ligase and 100 micromolars of Mg2+, the initial rate of ligation was 1.4 times 10 to the negative 6 ligation product per minute, while it was 3.5 times 10 to the negative 5th ligation product per minute in 100 micromolars of Fe2+. This is 25-fold higher in the Fe2+ than in Mg2+. For the hammerhead ribozyme, in 25 micromolars of Mg2+, the initial rate of cleavage was 0.011 fractions cleaved per minute, while it was 0.035 fractions cleaved per minute in 25 micromolars of Fe2+. This is 3 times higher in Fe2+ than in Mg2+. Finally, when these were tested in only sodium ions, the rates were 0, thus showing the efficacy of the control group.
Before the presence of oxygen, Fe2+ was accessible in a wide range of environments because it was not being oxidized. Also, Mg2+ was not very abundant because of the hypothermal systems associated with the higher heat flow and the sea floor spreading. Basically, water was not cycling Mg2+ very well, making it not very accessible to many environments. RNA with Fe2+ support more diverse RNA structure and catalytic functions than RNA with Mg2+. This is because Fe2+ actually pulls electrons off of the phosphate groups in RNA more effectively than magnesium ion.
Conversion of one metal to another is easy in some protein enzymes. This would make it plausible for organisms to switch from RNA to proteins and still survive when iron ion became unavailable during the Great Oxygenation event. RNA fragment that forms multiple first shell interactions with Mg doesn't change with iron-ion substitution. As stated in the Draper-Misra article, magnesium ion catalyzes RNA well because it is divalent, capable of making two bonds. Since iron is also divalent, the bonds with RNA are very similar to the magnesium RNA bonds. Iron ion substitute for magnesium ion to support catalysis by ribosomes. Ribozyme's activity when using iron ion is greatly enhanced compared to ribozyme activity using magnesium ions.
So in comparison, when RNA combines with Fe2+, it reacts more readily and efficiently than when it combines with Mg2+. In short, the above experiment and data presented show that RNA world hypothesis could be the possible origin of life.